Thank you for the introduction. My presentation will now cover the QISAR, or Quantitative Structure-Activity Relationship aspect of impurity classification under ICHM 7. This is an outline of my presentation. I'll begin with an introduction to how QISAR modeling works. I'll contrast the use of published structural alerts with QISAR model predictions. I'll list some of the QISAR software that are available for predicting the mutagenic potential of impurities. I'll cover the concept of applicability domain and also some structural limitations of the methodology. And then I'll move into the details of the ICHM 7 guideline and how QISAR should be used for this purpose. I'll also cover the application of expert knowledge. And then I'll finish up with some comments on comprehensive reporting of QISAR results, listing components of a report that are important. I'll provide an example format and also some special considerations with regards to software versions and also out-of-domain results. So let's begin with the basics of QISAR modeling. What is it? Well, QISAR stands for Quantitative Structure-Activity Relationship, and it's a type of modeling that identifies associations between attributes of chemical structures and biological activity. And in the context of this presentation, we're talking about toxicity, and more specifically, mutagenicity. It works under the general assumption that similar molecules exhibit similar chemical and biological properties, and therefore that toxicity can be explained by chemical structure. The modeling process itself learns from the results of actual laboratory testing. So we use a computer to examine a data set of chemicals that have already been tested in the assay that we're trying to build a model for, to identify pieces of those chemical structures that are statistically associated with a positive response in that assay. And those pieces are then labeled as structural alerts. The modeling process then goes a step further to look for additional attributes of molecules that, when occurring along with a structural alert, have the effect of mitigating its activity. Once we've extracted both these alerts and also mitigating patterns, we can use that information as a stored model to make a prediction of a new chemical's toxicity based solely on its chemical structure. And this gives us the ability to fill data gaps when empirical data are unavailable or inadequate for a chemical and we need to assess its mutagenic potential. And just to clarify this definition on my later slides, QISAR refers to a quantitative relationship that's most commonly seen in statistical-based models, whereas SAR refers to a qualitative relationship that's most commonly seen in expert rule-based models. But collectively, we refer to them as QISAR with the Q in parentheses. So what goes into building a QISAR model? Well, we really need three things. First of all, we need to have our set of chemical structures that we're going to learn from, and these need to be represented in a format that can be interpreted by a computer. And we do that through the use of molecular descriptors, which essentially serve as a fingerprint for each of the chemicals. And then for each chemical in the data set, we need to have a numerical score that denotes the activity of that chemical when it was tested in the assay that we're building a model for. So in the case of Ames mutagenicity, any molecule that was tested and shown to be a mutagen will be assigned a 1, and anything that was shown to be a non-mutagen will be assigned a 0. We then apply our QISAR algorithm or method for extracting those patterns, and we can pull out both the structural alerts as well as the mitigating features and store that information as our QISAR model. We can now come along with our new structure that we would like to make a prediction for, and the model will check for the presence of both the alerts and also mitigating features, and based on those, it will make a decision as to whether it considers that molecule to be either mutagenic or non-mutagenic. So in this case, the molecule was identified as having a primary aromatic amine as a structural alert, but also some mitigating features in the form of substituents on either side of that amine. And collectively, the model has made a decision based on that information that the molecule is likely to be non-mutagenic. QISAR methodologies can be loosely broken out into two categories: statistical-based models and expert rule-based models, and these definitions are used in the ICH M7 guideline. Statistical-based models are those that are constructed through machine learning, and they typically use a very large set of chemical structures that have been tested already, and the information that we provide the software when we're building the model is the chemical structure and a number which denotes its activity. The computer algorithm then extracts those statistical relationships between the chemical features and activity, and it algorithmically extracts structural alerts and mitigating patterns without any prior knowledge of mechanisms of reactivity. In contrast, expert rule-based models are those that are derived from human expert knowledge. They're generated through manual extraction of alert and mitigating patterns from known examples, and they have the ability to capture mechanistic information that may have been reported in the literature, or even based on the knowledge of the person who is defining the alert in the first place. They also have the ability to extract patterns from proprietary sources of information without those proprietary data needing to be in the model itself. The structural alerts and mitigating patterns are then encoded into the software so that they can be consistently applied by any user regardless of their level of knowledge of the endpoint. These are some examples of QISAR software that we've seen in regulatory submissions, and they cover both expert rule-based platforms as well as statistical-based platforms. And they're also a combination of commercially sourced models as well as freely sourced models, freely available models. The ones that I'm highlighting at the top in red are those that we use routinely at FDA CDER under research collaboration agreements with the respective companies. Applicability domain is an important consideration for a model that's being used for regulatory purposes. This defines a region of chemical space within which the model can make predictions with a given reliability. And that area of chemical space is defined by the structural attributes or the properties of the molecules that the model was trained from. So from a practical standpoint, this means that if your molecule, your impurity, is outside of that applicability domain, you can't use that model to make a prediction that we can use for decision making. Impurities that are outside of the model's applicability domain are sometimes referred to as out of domain or OOD, and they should not be predicted. But it's important to note that different models have different ways of interpreting applicability domain. And that comes from the fact that some of them are trained on different data, but also just they're perceiving structure in a different way. And so we can use that to our advantage to obtain a valid prediction, because we may find that one model is unable to make a prediction, but another model can be used and actually generate a prediction that we can use to support our impurity classification. However, when multiple models are all giving an out-of-domain result, then that's when we need to give that impurity extra attention. And it may be that QISAR is just not appropriate for predicting that impurity. And at that point, we may need to look to another way of getting information, such as an Ames test. The following is a compilation of structural classes that have been reported as being associated with mutagenicity in a number of leading publications that are referenced in the bottom right-hand corner of the slide. They cover molecules that have an aromatic motif, an aliphatic motif, or a halogen. And this type of a compilation can serve as a useful lookup tool for getting a general idea of whether a compound is likely to be mutagenic, but it's not a replacement for a QISAR prediction. So the question does come up, why don't we just rely on visual inspection? Why do we not simply use a lookup table to determine the mutagenic potential of an impurity? The answer to that is that the model can actually capture much more highly complex associations that are not captured by a general published structural alert, which doesn't consider mitigating features or the effect of multiple substituents on the impurity. I'd like to give an example of the primary aromatic amine to really illustrate this point. The amine is shown in the right-hand side of the slide, and the mutagenic potential of the nitrenium ion that forms, that then subsequently can form an adduct with DNA, is affected both by the electronic effect of the substituents in the R groups around the ring, but also based on the steric influences of those substituents. There's a publication by Alberg et al. That illustrates this nicely, and this is just a part of the table that appears in that publication, where you can see that there is a difference from strongly deactivating to strongly activating for the same substituent when it's placed in different positions around the ring. And so due to this complexity, I think it really underscores the value of using QISAR as a more refined approach to predicting activity based on all aspects of chemical structure. There are some structural limitations, though, to be aware of the QISAR methodology. It's really intended to make predictions for organic molecules that are on the lower end of the molecular weight range. When we get up to larger molecular weight molecules, such as polymers, plastics, proteins, polysaccharides, the model is not going to take into consideration higher level parameters that may affect its physical chemical properties, so for that reason would not be appropriate. Additionally, inorganics are generally not well predicted by QISAR models simply because they have much more complexity associated with different oxidation states around a metal and other properties that are not going to be well covered by the training set molecules. Organic chemicals that are mixtures cannot be predicted, but we can make a prediction for the individual components of a mixture if we know what they are. And of course, anything that's poorly covered, any type of molecule that is outside of the applicability domain due to perhaps some unknown molecular features, are not going to be able to be predicted. Lastly, but importantly, there is a limitation with regards to the way that the model interprets the structure in 2 dimensions. The model is looking at the connectivity around a certain atom rather than the actual configuration at a given atom. And so for that reason, it's not going to differentiate stereochemical pairs nor geometric isomeric pairs. So now let's move into the details of the ICHM 7 guidance and how we use QISAR in that context. Section 6 outlines how QISAR should be applied. It states that the models that are being used should be predictive of the outcome of the bacterial mutagenicity assay, so the AIMS assay. 2 prediction methodologies should be used. One should be expert rule-based and the second should be statistical-based. And furthermore, that the models being used should be consistent with the OECD's validation principles. It goes on to say that when those 2 methodologies have been applied, if there are no structural alerts that have been identified, then the impurity can be assigned to class 5 and no further testing is recommended. The OECD validation principles are a series of pieces of information that should be associated with any model that's being used for regulatory purposes. So there should be clarity around the endpoint that the model was trained on and also that it's predictive of. There should be clarity around the type of algorithm that was used. As a reviewer, we need to be able to tell whether the prediction is expert rule-based or statistical-based, and also knowing the algorithm gives us some insights into how to interpret the model output. There should be a defined domain of applicability to alert the user when the model just doesn't have enough information to make a valid prediction. There should be appropriate measures of goodness of fit, robustness, and predictivity. This speaks to adequate validation to show that the model performs well enough for this type of purpose. And lastly, it's ideal, but not necessary, to have a mechanistic interpretation of the model output, because that assists with our ability to understand and interpret the predictions. There's an important provision in the ICH M7 guideline for the application of expert knowledge to the predictions to the model output. And what this means is we can bring to bear additional supportive evidence on the relevance of any positive, negative, conflicting, or inconclusive prediction and provide a rationale to support the final conclusion. So if we disagree with the decision that the model has made and we have good evidence to suggest that it should be something different, we can provide that as justification for assigning that impurity to a different classification. So on the right-hand side of the slide are some examples of expert knowledge that can be applied, and I'm going to walk through them in the context of the impurity that I showed earlier in my presentation. Firstly, we can identify and interpret the alerting portion of the molecule and see if it makes sense from a mechanistic standpoint. So we see that the model has identified the primary aromatic amine as a structural alert, but we can also see that the model identified mitigating features in the form of substituents in the ortho position on either side of the amine. Mechanistically, we know this makes sense. The molecule is likely to be a mutagen through formation of the nitreniamine. However, if there are bulky substituents on either side, that's likely to reduce its ability to form an adduct with DNA. So this is a reasonable result from the model. We can then look at the training set structures that were used to derive both the alerts as well as the mitigating features to make sure that they are actually structurally similar to our impurity of interest. This is information that we can extract from the model output in some cases. But in cases where we feel that the model has not based its decision, its prediction on relevant information, we can actually bring in additional data for structurally similar compounds from supplemental databases. So these are data that were not used to train the model, and we can use that to either support confirming our prediction or overturning our prediction. And as a last point, FDA CDER applies expert knowledge to all QISAR analyses that are conducted in-house because we believe that it improves the overall predictive performance of the models. So now I'd like to talk about comprehensive reporting of QISAR results. Under the ICH M7 guideline, applicants can use any model that has been shown to be fit for purpose, and that could be a commercially available model, it could be a freely available model, or it could even be a model that was constructed in-house by an applicant. CDER has prior knowledge of several commercial and freely available QISAR models. However, in cases where we do not have knowledge of a model, we would require supporting documentation to demonstrate that that model is actually fit for purpose. And that would typically be in the form of a QISAR model reporting format or a QMRF. And I've provided some references at the bottom of the slide. With regards to actually reporting what was done for the QISAR analysis, there are several components that are important to include in such a report. There should be a materials and methods section that lists not only the software names, but the versions, because many of these software have been around for many, many years and the versions change over time. And so we need to know how current those models actually are. We like to see the individual model predictions as well as the overall classification, ideally in tabular format, because this is a very clear way to present the information. There should be a detailed explanation of any expert knowledge that was applied to the predictions, particularly if it was used to overturn the predictions. And what that means is if a structural alert was identified by one of the models and the impurity was assigned to class 5, there needs to be some explanation as to what information was used to make that final class 5 decision. And then ideally, we like to see an appendix containing the model output files, plus any other pivotal data. The model output files are just very helpful because they can allow us to see whether the models were interpreted appropriately, as well as other details that might otherwise not be available. This is a table of results just to show how the information can be presented in a clear way. It's color-coded for the purposes of this presentation. So I can walk through some of the example chemicals and how they reference one another. If we start with chemical 1, we can see that the expert rule-based as well as the statistical-based model identified the aromatic nitro group as structurally alerting. However, in the comments field, it's noted that this alert in the same chemical environment is actually present in chemical 4. And chemical 4 at the bottom was tested in the Ames assay and found to be negative. So for that reason, chemical 4 can be assigned to ICH-M7 class 5, so it can be treated as negative. And furthermore, that alert can be dismissed back in chemical 1 because the alert was present in the same chemical environment in chemical 4, and that was shown to be negative. So for that reason, now chemical 1 can be assigned to ICH-M7 class 4. Chemical 2 is a situation now where we have the corresponding amine rather than the nitro. The model did not consider that amine to be structurally alerting due to the presence of the other substituents in the alert environment and assigned it to class 5. And that makes sense. If we're going to call the nitro negative, we would expect the amine to be less mutagenic. So that would actually make sense from a mechanistic standpoint. And then lastly, chemical 3 is where we have that same nitro group, but now we're missing one of the R groups, which exposes the nitro and tends to increase its mutagenic potential. So in this case, we can't dismiss the alert using the data from chemical 4, and the positive prediction that's coming from the expert rule-based platform still stands and leads to that impurity being assigned to class 3. And I should also point out in that case, one of the models, the statistical-based model, was unable to make a prediction, but that doesn't matter at that point because we're going to go with the positive from the expert rule-based platform. There are a couple of special considerations that one should be aware of with respect to QISAR results. The first is with regard to the version of the software that's being used. Predictions generated with the most recent version of a software are preferred, but old predictions may be acceptable unless the conclusions are questionable, and this could be in a case where you have a negative prediction for a chemical that has a visual structural alert. That may be where we look a little more closely or possibly even rerun a prediction. Internally at FDA CDER, we consider QISAR predictions to have about a 2-year shelf life as an unofficial rule of thumb. And this reflects the fact that the models are updated about every 1 to 3 years. And so after a couple of years, we may be worried that the predictions are out of date. And so it's important for us to be basing our decisions on the most current information. So if we receive a submission with predictions that are older than that, then we would want to look at them a little more closely. Out-of-domain results are another area that warrants special consideration. An out-of-domain result is not a prediction. It's when the impurity is outside of the applicability domain of the model. So it does not contribute to a negative or class 5 assignment. And what that means is if you have a negative result from one of your models and an out-of-domain result from the other model, you can't look at that combination of results and say that there are no structural alerts and therefore it's justified assigning the impurity to class 5. This is a case where you actually have an incomplete analysis. So that data gap can be addressed through the use of expert knowledge to resolve that lack of information from the second model, or it could be addressed by running another model that can actually give a prediction so that you have that complete analysis. And we've generally found that that method with the third model works well for us at CDER. So in conclusion, QISAR models provide a state-of-the-art approach for assessing mutagenicity. The prediction is based solely on chemical structure. It replaces visual inspection as a way to identify structural alerts for impurity classification. Published structural alerts can have value as a preliminary screen for potential mutagens, but QISAR is needed for a more refined prediction. Any models that are being used should be consistent with the OECD validation principles. Comprehensive reporting of impurity analyses really helps avoid multiple review cycles, so it's important to do that up front. And lastly, FDA CDER applies expert knowledge to all internal QISAR assessments because it's something we believe strongly in. And with that, I'd like to acknowledge the members of our internal computational toxicology consultation service team, as well as internal collaborators at CDER and our external collaborators who provide the QISAR software that we use. And then finish up by saying that I'll be happy to take questions in the panel discussion at the end, but additional questions can also be sent to the following email for follow-up. And I'd like to cross-reference this presentation with the one given by Barbara Scott to give a more broad overview on impurity classification. Thank you for your attention.